In this video, we're going to continue our discussion on controlling microbial growth, and we're going to concentrate in this video on the chemical methods used in medicine and industries, etc., for controlling growth. So just to go back in and refresh our memory on some of the terminology, when we say sterilization methods are going to ensure the complete elimination of all microbes to include the endospores and deactivation of viruses. You may remember in our previous video with the physical methods, we talked about autoclaving and those gamma X-rays for ionizing radiation. Now, when we talk about disinfectants, we're going to see that they are going to be a physical process. And in this video in particular, there's going to be lots of the chemical agents which are going to be used that are disinfectant, and they're going to be able to destroy the pathogen and even destroy the toxins produced. Antiseptic, again, are going to be chemical in nature, so we're going to look at several in this video today, and they're going to be able to destroy or inhibit those vegetative or living pathogens that are on living skin. Now, when we say living skin, we're talking about the tissue that is below that outer layer. So it has to be living tissue, so the deeper layers of skin and the mucous membranes. And that'll come important here in a little bit. Sanitation, we're just reducing or lowering the numbers, and decontamination or degermination is that mechanical removal. So we've already talked about the physical control. In this video, we're going to look at chemical control, as I've already stated. And why? Because we need to try to find ways to ensure that we can prevent diseases, that we can help with the food preservation and other industrial applications. So we're going to use the chart that's in your lecture notes so that you can follow along. Now, before I begin, I'm going to go ahead and answer that question. What do we have to know out of all these charts? So for each of the agents, these are all chemical agents. What you need to know in particular is what's the level of control. I want you to know the examples that are provided in there, especially when they're name brands like Clorox and Lysol and things along those lines. And then the very basic mode of action. So even if I give a little bit of specifics, I'll try to reel it back in and just say this is what you need to know as far as that mode of action. So let's begin with our phenolic compounds. These are going to be chemical compounds. They're going to disrupt microbial cell membranes. They're going to also precipitate, if you will, so clump together proteins. In between disrupting the cell walls, the membranes, and precipitate proteins, they're going to be able to shut down cell metabolism and ultimately lead to that microbial death. Now, when we talk about phenolic compounds, Lysol is the main example. It contains phenolic compound like the orthophenylphenol, and that's going to be used on a wide range of items found in everything from hospital cleaning agents to bathroom agents, etc. There's also another component known as trichlorosan, and that is used in soaps and toothpaste, cat litters, deodorants, and it's another phenolic compound. All phenolic compounds, though, for the most part, are going to be used as disinfectants. So the level of control is a disinfectant. Now, let's talk about alcohol. Alcohol, either ethanol, which is ethyl alcohol, or isopropyl alcohol, they're widely used in all areas from skin all the way to surface cleanings. Now, alcohol works because it's going to go in and dissolve that cell membrane by reducing that surface tension. Think about alcohols and how they work, and if it gets onto those cell membranes, it's literally going to melt them away. So therefore, it's going to be able to, if you destroy that cell membrane, you're going to be able to kill the cells. Now, one of the misnomers about alcohol is that if it's 100%, it's going to be more effective than those that you see at 70%, etc. Nope. Actually, 70% is one of the most effective. So it has to be at least 50% or higher, but it can't be 100% for it to be an effective disinfectant. So we all have various examples of how various alcohols are used, from cleaning wipes and things that we use them from just out of the bottle itself for a surface cleaner. Now, one of the things that I want to clarify, if you look on the web, it's going to say, hey, alcohol is both a disinfectant and an antiseptic. Now, as just mentioned when we were talking about antiseptics and going through that definition again, antiseptic means that it can be used on a living tissue such as skin and mucous membranes. Now, when do we use alcohol? We use alcohol wipes to wipe on the outer skin before getting shots, before getting blood drawn, etc. Well, just go back and remember your A&P for a minute. That outer layer of epidermal tissue is not living tissue. That is where all the dead cells have moved up. So when you touch your actual outer layer of skin, what you're touching is dead tissue, not living tissue. If you cut yourself and you expose the living tissue beneath it, should you pour alcohol in an open wound? No. For many reasons, but because of the fact of how it works by dissolving the cell membrane, it can actually fuse those cells together and wall it off and actually cause more harm than good. So because you should not use alcohol on an opened wound in the living tissue, I say for this class, it is a disinfectant only. Why? That helps you keep the terms true. All right, so for us, alcohol is a disinfectant. When you use it on your skin before a shot or before drawing blood, it's a degermination process. You're literally physically wiping down and removing microbes, and it might kill anything that's living on the outer tissue. But remember, again, I'm going to say it for the third time, that outer skin layer of epidermis is non-living tissue. So to be in the pure term and definition of an antiseptic, we don't use alcohol on living tissue. Okay. Back to our list, if you will. So the halogens get their name because of the halogen group in the periodic table. It's going to contain chlorines, fluorines, iodines. They are very powerful disinfectants and are used quite frequently as water purifiers. So how do they work? They work by disrupting proteins and enzymes, which then that means they're going to disrupt metabolic processes. They are considered to be disinfectant with the exception of betadine, which is going to be an antiseptic. So let's look at some examples. Chlorine, hypochlorite compounds, those are going to be used in household bleaches such as Clorox. They're going to be used for chlorination of pools and spas. Fluorine is another one that is added into drinking water. And then iodine is commonly used as an antiseptic in the form of that iodine tincture or betadine, if you will. So hydrogen peroxide. The example for this is hydrogen peroxide that you think of in the brown bottles, etc. And we're going to use it on wounds and various things. However, one of the things that I want to point out is that when you go buy hydrogen peroxide at the store, what we're buying is a 3% hydrogen peroxide. And at 3%, it is going to be considered an antiseptic and can be used on mucous membranes and living tissues. So hydrogen peroxide at 3% is a great antiseptic. However, you can also get hydrogen peroxide if you are buying it for lab uses and things like that at a higher level, and it is a great disinfectant when you can get it at the higher percentages. Now, how hydrogen peroxide works, no matter what its percentage, is the fact that it's an oxidizer, and it takes the oxygen and it converts oxygen into toxic forms. And those toxic forms of oxygen damage proteins and lipids, and you know if you damage the proteins and lipids that you're going to disrupt membranes, metabolic processes, and that can lead to cell death. Heavy metals. When we talk about heavy metals, it's just like it sounds. So it's going to be those metals such as silver and copper. And when you look at them, those are going to have antimicrobial properties. How? Because they have what we call an oligodynamic effect. And what that does is it binds to proteins at their active sites. So then if you block their active sites, proteins are not able to go in and then attach to their targets, so they inactivate proteins. Where that becomes essential is in enzymatic processes. If you shut down the enzymes within a cell, you're going to disrupt metabolism, and then that's going to lead to cellular death. Now, these heavy metals are going to be used in like cosmetics and ointments and eye drops. In particular, they're going to be incorporated in wound dressings, and silver ions, for example, can be found in silver, what they call the sulfur iodide creams that are used for burn treatment. So lots of different uses for the heavy metals, and they're going to be used as disinfectants generally. Now, gases. Gases are essential because of the fact that they're needed for sterilizing medical equipment and other sensitive materials, that some of these other strong chemicals aren't effective, or even in some of those physical methods that we talked about in the previous slide, such as autoclaving and dry oven and things like that. So think about plastic materials and some sensitive medical equipment, or just even containers that are plastic. You got to find a way to be able to sterilize or at least disinfect them, and that's why we use gases. One of the most popular is the ethylene oxide gas, and so that is the name that you need to remember for that. And what happens with the gases, and when you're using ethylene oxide, it's going to go in and disrupt the DNA and protein functional groups on them. So now again, that's going to lead to ultimately cellular death. Now, talking about soaps and detergents. When you talk about a soap or detergent and it is a pure soap, then it is only going to be a decontamination or a degermer. Why? Because soap itself is a surfactant, and it's going to disrupt the cell membrane, cause it to stick, and it's going to be able to physically remove microbes from the surfaces. Same thing with detergents. However, I know you're right now going, but my soap says it kills 99% of germs, or that it's killing 99% of bacteria, etc. Well, when you have a soap or detergent that says it's killing microbes or has an antimicrobial effect, that's because of the chemical compound that may be added to it. So it may be a heavy metal, it may be a halogen, but there's been another chemical compound added to the soap that gives it that antimicrobial effect. Soap on itself is a decontamination method because of its surfactant abilities. Talking about alkylators now. Now, alkylators, because that's alkaline or kind of groups of chemical groups within them, they are able to damage protein and DNA because of the way that they cross-link carbon sources because of being an alkaline type products. Now, where these are going to be used most likely are in formaldehyde and glutaraldehyde. You've heard of those when you think of embalming and agents and things like that. You know the strong smell that comes with formaldehyde, for instance, and so they're not used. Formaldehyde is not used very often anymore because it does have that highly noxious gaseous odor about it. And in fact, they have found that formaldehydes cannot, with heavy exposure, can actually be carcinogen. So now more in lab settings and things, they're using the glutaraldehyde, which is not a carcinogenic product, but it still has that noxious agent. So, but they are sterilizing if used at the proper concentration, but generally you'll find that they are considered to be disinfectants. An alkaline product as well is that ethylene oxide that we talked about just a moment ago, that is used and added to a gas solution to help with that sterilization product. And the last is acetic acid. I added this one because what it does is it goes in and adjusts pHs, and it can destroy or inhibit cell membranes. And I wanted us to have one that was kind of purely for the food industry, and acetic acid is a pickling agent. So it's often used to help us preserve foods, and it can help to decontaminate as well. So again, these are the chemical agents used for helping us to control microbes. By no means was this the whole list. We couldn't get through it, but these are the ones I wanted us to concentrate on for class. So know the name of the agent, be able to identify those examples provided, know the level of control that it provides, and then know at a high level what that mode of action is. If you have any questions, just let me know.